Matt, thank you so much for offering to do this session for us. We were so excited when you approached us because we're always trying to bring different and exciting topics to not just our clients here at the Tech Park Incubator, but also the community. Just a little bit about me. I'm a director at University of Arizona's first Center for Innovation, mouthful, and we are a startup incubator supporting startup companies based on science and technology. We're growth-oriented companies that can really make a difference in the world sometimes, a lot of times in the US, but really using technology and science to scale up business opportunities to address real problems. And that is where I really met Matt, as we've been working on our project up in Oro Valley, where we'll be building another incubator. And Matt, as soon as he came to town, I think pretty much was involved with that, working at Bayer, managing the site up in the northern end of our Southern Arizona region. And again, Matt has such a grand and extensive background that he wanted to share, and we welcome this platform. So with that, I'll just hand it over to you, Matt, and maybe you can talk a little bit more about yourself.
Sure.
And also get going on this particular session. Thank you so much.
Yeah, thanks so much, Anita, for the invitation. And thanks everybody for calling in today. I haven't really ever tried to do anything like this before, and it's really because of the kind of strange environment we all find ourselves in with COVID that it even occurred to me to start talking to Anita about doing something like this. And so, you know, I'd love some feedback and input at the end of this, if this is something you guys found valuable or not. But the basic idea was that agriculture is a place that's a really booming technology, or it's undergoing a real renaissance in technology and technology adoption. And for the last 15 years or so, we've seen a really huge uptick in the types of automation, the types of engineering, et cetera, that we're doing. And agriculture approaches these things from a really different point of view than a lot of people do, because of the kind of the foundation that it's built on, and both from a plant breeding side as well as from a, you know, roots deep in agriculture. And so I thought I'd talk about that a little bit today. This is going to be more story driven than science and technology driven. And that's part of why this is an interesting talk for me, because I'm a scientist by background. My PhD is from Arizona State University in plant cell biology. And then I went to Rice University in Houston to do some postdoc work before I settled with what was first, I was hired by Monsanto and worked for Monsanto for 9 years. And then, as a lot of people know, Bayer acquired Monsanto about 2 years ago. And that I've been with Bayer ever since. And so I've been a scientist my whole career. I'm a scientist today. And what I've focused on has changed over time. And that's, I will kind of weave that through what we talk about throughout. I'll try to leave about 20 minutes at the end for questions. And we'll see where it goes from there. Let's see. Okay, so every talk I give, one of the joys of working for a large multinational corporation is that we have very well-paid lawyers that help us make sure we stay legal in what we do. And so every talk I give, I get to put our legal disclaimer out. And it essentially says that I will talk about the future in this talk and some predictions, just because that's the nature of thinking about science and technology. I'm no better than anybody else at that, which means I'll be wrong more often than I'm right about the things I'm talking about. And you can't hold me or Bayer legally liable for being wrong about the future. So before I dive into who I am and what we're doing, I wanted to just cover Bayer real quickly. And Bayer is a company of about 105,000 people and has three major divisions: pharmaceutical division, which makes a number of prescription drugs; our consumer health division, which we're probably most famous for aspirin, you know, what we all take when we feel a little bit achy, or for blood pressure. And then our crop science division, which Bayer has had a very, very strong portfolio in crop protection chemicals for many, many years. Monsanto, and Monsanto was acquired, brought a very robust seed business with it, so corn seed, soy seed, et cetera. Part of why I think it's really important to show this slide right now is because in this world where we're all trying to figure out how to most effectively respond to COVID, COVID touches all three of our major business areas. So pharmaceuticals and consumer health, no doubt about it, and the way we think about healthcare. Crop science and making sure that we keep our global infrastructure for producing food in good shape, as we don't want to see a pandemic crisis followed by a famine crisis if we don't pay good attention to that. And so all three of our areas are business critical, and we've at some level tried to keep everybody operating through the last couple months. And then just real quick, why we think about technology in agriculture. This is the slide that we all kind of, everybody, whether you're an academic or an industry person, you tend to give this slide, and that's that we know the population's growing. There's some mega trends that we can see when we look until 2050. Another 2.2 billion people coming to dinner every night, and not only that, they're going to eat more meat by and large, and so that means that we need actually 50% more food to produce the calories that everybody wants to consume up through now until 2050. And that's not a simple problem to solve, and that's something that it's one of the things that gets me out of bed every day, is thinking about what part of that puzzle can I help contribute to on any given day. And also a lot of pressure on our ecosystems. We're seeing climate change start to have a real and present effect on harvests. And so how do we combat that using a combination of digital and biological and engineering solutions? And then just the general trend and loss of arable land per capita, both from construction and from other reasons. And so we need to help secure a sufficient supply of quality food. And then it's incumbent upon us to make sure that we use our natural resources efficiently and responsibly, with not only within Bayer, but helping our customers in agriculture do that. And the science of, or the mission of Bayer really is one that resonates for me as a scientist. Our foundation is science for a better life. Our mission is science for a better life. And everything we do has a basis in science. And I hope you'll see a little of that woven through the rest of the talk today. And so just to touch briefly then on the crop sciences division, the areas we're focused on is delivering world-class innovation, so really thinking differently about how we approach agriculture. A big part of that is our digital transformation. So there's a ton of effort and energy going into startups in agriculture right now, and a big chunk of that is digital in nature. So how do we use things like data about the weather to make better predictions about what we should be planting that year? And then setting new standards in sustainability, not just helping farmers use less water, use less fertilizers, but actually dipping a toe into regenerative agriculture. So agriculture is a net positive in the way we think about sustainability. And then driving to operational excellence, which I'll talk quite a bit about today. And so that within, on the right-hand side of the slide, then within crop science, that means we focus on seeds and traits, so selling seeds to farmers. And a lot of those seeds have biotech traits or native traits for things like disease resistance. Crop protection, so things like insecticides and herbicides, and digital farming. And those all come together, and there's this trend in agriculture towards tailored solutions. So it's solutions that are specific to each customer, not a solution that every customer sort of pulls from. All right, and then what are we doing here in Tucson? So 4 years ago, Bayer had no presence in Tucson. Our R&D division decided to make its largest investment in R&D, capital R&D, in the last 20 years by building a greenhouse in Marana. So I'm somewhere between 30 and 45 minutes from where most people live in Tucson, just west of I-10 in Marana, near the Marana Airport or Marana High School. What we have is 7 acres of a greenhouse space plus another 2 acres of automation space. So that's the 7 acres are split into 3.5 acre parcels, and then the automation bit. And why this is really important for us is this 7 acres takes the place of about 190 field acres of corn when we take in all the technologies that we're bringing to bear on our greenhouse in Marana. From genetic testing that we do that allows us to do selections in a laboratory instead of having to go out into a field for physical observation, to the efficiencies we gain by being indoors and protected from the elements and from pests, et cetera. And so my career at Bayer, the 11 years that I've spent at Bayer, has at least 5 or 6 of those have been focused on figuring out how we bring the technologies that we need to Marana to make this facility operational. And so I'll spend some time talking about that today. And then I did want to just pause for a moment on why am I giving this talk in particular? What reason do I have to speak about COVID-19, and why would I be talking to people at the tech parks thinking about startups? And part of it's that social responsibility part. COVID-19 is here, and it's going to have lasting effects on the way not only we operate, but the way we even think about innovation. And I wanted to share a little bit of what that's looked like for Bayer when we think about innovation. And I've always found when we're trying to solve difficult problems, laying different flavors of solutions alongside each other in non-obvious ways can lead to really interesting ways of approaching and dealing with difficult issues. Arizona, frankly, also is lagging in COVID testing per capita. We're, depending on the day or the week, we're either last or within the bottom 5 of states in the US per capita. We have a real testing gap, and that makes it difficult for employers to know how to safely reopen their sites for operation, myself included. It also makes it difficult for us to know how aggressively to scale our responses. And that's really, for me, it's something that I have a real passion around, because the other half of my career with Bayer has really been focused on genetic testing. So doing DNA tests to accelerate breeding. It's a very similar suite of technologies to what would be used for COVID testing. And at heart, I'm a technologist, and I really believe technology can help us solve our problems. And so, how do we think about that, and how do we approach that? And agriculture, I'd say, within the suite of life sciences technologies, is better than anybody else at testing lots of things really cheaply and really quickly. And I'll speak a little bit about why that is. But before that, a couple of caveats. So we're not going to change our operations in Tucson. We're going to continue to focus on food. And I mentioned why that's such an important part of our mission within this environment. And so as much as I'd love to jump in with both feet and figure out how to solve this testing problem myself, that's not something I can do. It's given the mission that we have. I also don't know anything about small business. I've worked for Bayer my whole career. I've been in industry. And so I fully appreciate that you're going to hear some stuff and think that this guy has no idea what he's talking about when it comes to small business. And that's true. So just take with a grain of salt anything you hear, and kind of take what's good and reject the rest. And I know nothing about, nothing other than what one could easily read in an article about medical testing. And there's really different constraints around how one does medical testing from how one does testing for crop breeding. And so I appreciate not all the things I talk about will be super relevant, but knowing what little I know, I tried to make this as relevant as I could. And then the picture on the right is just part of why I'm giving this talk, is because we are a member of this community. So this is where our greenhouse is right by Pitcher Rocks. University of Arizona is over here, and the tech parks at Rita Road are over here. And so we are in Tucson, and it's very important to me that we all figure out how we come through this together, and to my team as well. So this is now the little bit where I talk about why I think I have some relevant experience. And so I think a lot about crop breeding, corn breeding in particular. And breeding is the gradual process of changing a plant to something that humans like a little bit better. And so when you think about the ancestor of corn, that was a little tiny 6-inch long ear that came from a plant called teosinte. And that plant wasn't edible. It was edible and people ate it, but it was difficult to eat. It looked nothing like the corn on the cob you'd eat for dinner. It had these armor-plated kernels that had to be laboriously cracked open. The yields were extremely small. And basically, if you were a farmer, you spent your whole life farming. You didn't get to do anything else. And everybody spent their life farming because it was so difficult to produce food. Over years, over millennia, in the case of the conversion of teosinte to corn, farmers and breeders identified 6 natural mutations that changed teosinte from that little puny 6-inch long cob into something that looks a lot more like what you would eat for corn on the cob for dinner at the dinner table, or what a farmer would harvest as field corn. And so big, huge change, or some breeding leads to big step changes in how a crop behaves, and some are just small incremental changes, small boosts in yield, small changes that make it easier for the breeder, the farmer, to process the crop. But I do want to really emphasize, and I'll probably touch base on this a couple times, the breeding that we do at Bayer today, the breeding that any breeder does at a university or any of our other competitors' companies, is using the same philosophies, the same science that's been in play for hundreds and thousands of years. Just the tools are getting a lot more sophisticated, and we're starting to scale so that we do lots and lots really quickly. And so what that looks like to the modern breeder is it's got 2 components, a lab component and a field component. I've spent my entire career on the lab side of this, and this journey into the greenhouse that I've been doing for the last 2 years is the thing that's maybe going to help me bridge that space into the field a little more. Worked in a lab where we did seed chipping so that we could take little small samples of seeds. We could extract DNA and do DNA testing on those. We test hundreds of millions of those seeds a year, and each one of those seeds will do between 1 and hundreds of DNA tests on them using lab automation. That generates a ton of data, and data, when you automate, is as big a problem as the physical engineering of the automation. And so then every night we're processing those hundreds of thousands of records so that we can make decisions, throw away the trash, so the things that aren't going to look good in the field, and then keep the best. And then because of the investment in testing, so we've spent now money, and that's, no matter how cheap it is, it's still not free. And because we've injured the seed by taking that little DNA sample, when we put those seeds in the field, they tend to be pretty wussy and they tend to get eaten up by bugs, blown over by wind, et cetera. And that first generation post-lab, we want to put them in a greenhouse. I want to re-emphasize here, when I say lab, I'm not talking about biotechnology here. I'm not talking about making a GMO. I'm talking about just the genetic testing that used to all be done by manual observation in a field. We can now use the genetic sequence of a seed to predict how something's going to perform in a field to some extent. However, labs and greenhouses are still not a perfect replica of what we see in the field. And so then we have an extensive field testing phase that our crops will go through before we're willing to sell it to a customer. And then this is drawn as a cycle because this goes round and round and round. It takes, on average, about 10 to 15 years to generate a new variety of corn, starting from year one. And our greenhouse is going to touch that at 2 points in its life cycle, in about the first 5 years and then about the last 15 years. Okay, so me, and a little bit of my background. These are the 4 major areas I've worked in since coming to Bayer. So high throughput, so I was hired as I mentioned my PhD was in plant cell biology. I was hired as a plant cell biologist. And rapidly, so for anybody who has lab experience, most of what you do in a lab is you have a thing called a pipetter that's looks like a little pen. And you move very tiny volumes of liquid from one small container to another. And you do this over and over again, and it's super repetitive and mind-numbingly boring. And I realized pretty quickly after starting at Bayer that this wasn't a career for me. I wasn't going to be able to make it. I'd done it for 6 years in grad school and postdoc, and I was just losing my mind in my first year at Bayer. Now, fortunately, robots to the rescue. So robots are quite good at this, and they don't get bored as long as you program them effectively. And so I got involved in a lot of automation projects to not only alleviate my boredom, but then to really increase our capacity for doing cell-based assays. So doing imaging, testing how GMOs perform inside of cells, et cetera. That bridged me into a space where I understood the automation for laboratories really well, the same automation that was used in our high throughput genotyping labs that I've already talked about a little bit. And so I took over leadership of the group that developed all the new workflows for genetic testing. So new molecular assays, new automation. And I led that team for about 2.5 years, and we've worked on a number of projects to accelerate and expand our genotyping capacity. In parallel, did a really interesting project that was completely different in scale. It was a point of sample genotyping. So figuring out where we have samples in a very low throughput way, can we get good information about the genetics of soybeans in this case. And this is a picture of soy for those who haven't had the joy of meeting a soybean face to face. And then from there, I stepped into so using my understanding not only of automation, but of how we think about deploying new technologies, took on this role here in Tucson, leading the greenhouse project and the deployment of our greenhouse, which is a fully automated greenhouse. It's actually about a 9-acre robot when you look at all the different components that we have that are interwoven throughout our whole site. And so the one thing that I learned as I've done all of this is that ideas are plentiful. So it's really easy to come up with different ingenious ways of doing things. But scaling them is always hard. There's never a thing that we've done where it wasn't much harder to go from doing it 10 times a day to doing it 100,000 times a day than it was to come up with the original idea itself. On average, it takes us 6 to 12 months to come up with a really good new idea, to vet it, to compare it, to figure out if it's the thing we want to do. And then it takes us another 2 to 4 years to deploy it at scale in a way that can fundamentally accelerate the way we think about breeding. And there was a rough moment in my career where I had to kind of look in the mirror and understand that I'm actually pretty bad at the ideas part. And that for a guy who had spent most of his formative years as an academic, really driving science, admitting to myself that coming up with new ideas wasn't my strength was tough. But it came in parallel with the realization that what I am really good at is the scaling of ideas. And so I can talk to people who have lots of good ideas, help them figure out which ones are scalable, and then get those into what I would call a production environment. And so in a place where we can do 10 to 100,000 things a day or a week repeatedly. But then I'm also exquisitely bad at doing the same thing every day for the rest of my life. And so it's that sweet spot for me between the coming up with the new ideas and the doing it every day, where how do we take interesting ideas and how do we scale them to best effect? And that's been about 17 years of my career, both within academia and at Bayer. And so at Bear, and I think this is generally true of a lot of different workflows, but our workflows are conceptually simple. When you lay the steps out, it's really not that difficult to do. So I've got two different workflows shown here. The DNA isolation and detection workflow that I did on the genetic testing side of things, and then what we do in our greenhouse on the bottom. And so starting at the top with our with DNA, we take those seeds and we invented seed chipping technology. And so we literally take a seed, we use a robot gripper to pick it up, and we take a saw blade or a drill bit and we take a chip out of the top of the seed. The top part, that's all that juicy endosperm part that when you bite corn on the cob, that's what kind of squirts out. The corn seed is actually okay if you take a little bit of that away. Now the embryo is at the bottom, so this is a little baby corn plant. If you chip that, then it's dead. And so part of what we had to learn to do is orient those seeds appropriately, so we didn't kill them every time, but we can test hundreds of thousands of, we can chip and test hundreds of thousands of seeds a year. And the workflow is very simple. We sample, we extract DNA, we detect DNA, and then we analyze and select. It's the type of thing where when I was a postdoc, my 2-year-old came and sat on my lab bench, or in my chair next to my lab bench, and he would occasionally help me with some of this. Like monkeys could really do it if we train them appropriately once or twice. What gets really difficult is doing it 100,000 times. And that's, to me, what just gets me really excited. And I've given some examples of the different types of things for anybody who's knowledgeable of the type of technology space I work in, the different ways we think about DNA extraction, the different ways we think about DNA detection, technologies that are anywhere from 20 years old to technologies that have been deployed within the last 3 years. And at times we've been the first people to ever deploy that type of technology at the scale that we're thinking about. And then doing those selections, there's no point in doing all that testing if we don't gain efficiency. And so selecting in a lab and then sending to our greenhouse. And then our greenhouse, dead simple. This is even easier for my kids to do. We do this in our garden frequently. So we plant the seed. We need to transplant it. So we take it from the little pot, we transplant it and put it in a 2-gallon pot. Somebody's got to pollinate those to make sure that the right genetics go to the right place, and then somebody's got to harvest it. This is, again, dead simple. But doing it 10 times is a very, very different story than doing it 120,000 times in a year. So our scale is not quite as big as the DNA sampling scale, but again, scale really, really matters when we think about technology. And it really drives me when I think about technologies to choose the simplest technologies I possibly can if I'm going to be scaling them, because as soon as you multiply, it gets hard, really fast. So one of the points I like to make when I talk about genetic testing in particular, and so again, going back to COVID a little bit, when I say genetic testing, I mean we isolate DNA, we amplify specific portions of DNA, and then we do a diagnostic on that to ask what's the flavor of DNA at that spot. And then we can look across entire genomes, and that's what these little cartoons here show us. This is half the human genome. This is one of the 10 chromosomes from corn. We can look very deep inside of a genome and use that DNA, that little spot of DNA, to predict things about the future, about a health condition. And so for humans and for plants, we need to do DNA analysis. COVID's not DNA per se, COVID's RNA, and there's a little distinction there, just in the flavor, what the DNA is, and the way you isolate it, and the way you amplify it is slightly different. But by and large, you're talking about a similar suite of techniques. RNA is harder to work with than DNA, no doubt about it. What's kind of fun about plants and what's different between plants and people is there's a couple fundamental differences. In people, you don't get to set up your population. So it's really frowned upon to breed humans for the sake of understanding their genetics. This isn't a thing that this is something nobody ever should or will do. Plants, we do it all the time. So we can set up our populations in really intentional ways to make sure that we can infer the most information possible out of plants. And so that's thing one that's really fundamentally different about the way we think about genetic testing in plants and in humans. The second is what you're informing. So in humans, you're really informing a diagnosis. You want to diagnose diseases, save lives, improve quality of life. And so because of that, there's functionally not a real upper limit on what we're willing to spend for a lot of these tests. We'll spend $40,000, $50,000, $2,000, $10,000, $20,000 per sample if it's going to make a difference in somebody's life. Plants, it's totally the opposite. I'm going to start with 1 million corn plants. I want to throw away 900,000 of those because they're all losers. They're all not quite as good as the 100,000 I want to leave behind. And so I only want to get enough information to figure out who's terrible. I'm not trying to get enough information to figure out who's amazing. And that's a fundamental difference in the way we think about plants versus the way we think about people. And what that means from a technology point of view is that I want the crudest, cheapest, fastest, highest throughput technologies possible because I have huge populations that I'm dealing with. And I don't need very much information to make decisions. I don't do any paperwork when I throw a plant away. So even if I make a little bit of a mistake, there's no review board, there's nothing that happens that makes my life difficult. And so fast, cheap, efficient is the name of the game. And so we use the same technologies. I use the same sequencer to sequence DNA that somebody's using in a hospital today. I use the same PCR machines. In fact, we're looking at donating some of our PCR machines to testing labs in places like St. Louis right now, because it's really difficult to set up to convert a lab from plant testing to human testing just from a regulation point of view, but the equipment's so similar that we can donate the equipment for people to use to expand their testing capacity. So it's very similar, very identical technologies, really different applications. And that means everything when it comes to how we think about scaling. I will point out though, there was this mind-blowing moment I had when I was at Washington University in St. Louis. And I was listening to a woman who did research on malaria, talk about the work she was doing in Africa. And she laid out the same exact problem that I just laid out for you guys. And so there are some applications in health where cheap, fast, and cheerful would fundamentally change the way that we approach solving problems. And I think it's becoming clear that COVID is potentially one of these as well to me. And so with that, that's the majority of the background. And I have, I think, another 6 or 8 slides. I just talk about a couple lessons I've learned that I'll share with a couple of examples to sprinkle in. And you know, again, this is more kind of food for thought. There's a lot more behind this than what I can share in an hour. And I think there's opportunities if we need to take a deeper dive in some places to talk. But I wanted to talk first about just, you do invest. So to scale, you have to spend money to do it. It's expensive and it's difficult. And so what we've gotten really good at is investing in what pays for itself. So we only want to invest something. So if I invest $500,000 in a new robot, that needs to make me $500 to $1 million in saving. Because we're in R&D and we don't have customers, that's in savings. So operational savings typically for us. But we spend a lot of time upfront doing math. What's the opportunity cost here? And if we were to apply technology, what would we save? What would we gain? And usually the way we think about that is we're trying to establish a technology baseline or a technology floor. And so we take, every time we deploy a fundamental new technology, that becomes a new floor that's a platform for innovating upon. And so things that we didn't think would be possible 5 years later, we are only able to do because of new technology we put in place 5 years before. And I'll give a couple of examples of that as I go. The graph on the right, because some of the information is proprietary on this, I can't show the scaling on the y-axis here, but the x-axis shows years, and what you can see in the bars is the total data points we're running. And I can verbally say that I think 2018 was the year we decided we had done 1 billion data points in our labs that we were running. And so we've, over a 20-year period, we've scaled our throughput massively for all of our genetic testing. This is again, genetic testing, not greenhouse stuff. And along the way, we've dropped our cost per data point. And the goal all along has been, how can we do more for the same amount of money? So if you were to see a line that showed operational cost, it's stayed somewhat flat, if not gone down over time. And so what we've had to do is figure out how to do more with the same budget year over year over year. Because when we look at 1998, it was super duper expensive to do a handful of genetic tests. It was so stupidly expensive that we would never contemplate doing the things that we're doing today in 2020. Because in 2020, well, and so there's about 3 or 4 generations of lab automation that baked into this 20-year period. And each generation became a platform that allowed us to think about what would be coming next. And so when you're at a price point where it's $20 or $50 a sample, you're going to think that maybe, you know, a good, well thought out experiment or diagnosis, you've got a couple of, you know, thousands of dollars to tens of thousands max per experiment to do. And so in the early days, we just did small experiments, small tests. But as it became possible, you know, let's say we hit $10 a sample here in 2013, all of a sudden we've increased by an order of magnitude the number of samples we can do for the same cost. And then you can kind of see how that changes. We structure the way we approach science differently now because it costs so little to get a data point. And so we're now using data for breeding because we had to hit an inflection point. I think we hit it about 2016 where data in a lab could take the place of physical observations in a field. It required this number of data points to be able to do that. Before that, the physical observations were far superior to the data predictions. But once we make that inflection point, once we get into the lab, all of a sudden we can scale the size of the populations we test ad infinitum, almost limitlessly, because we're not talking about field testing, which field testing is hundreds of thousands of acres where we have crops growing out in the field that we have to do observations on. Doing it in a lab lets us take advantage of economy of scale. I won't touch as much on this because it's not my area, but I always make sure I've got good data people whenever we're talking about scaling automation, because with more samples comes more data, and with more data, more problems in a lot of ways. Once you've solved the data problems, they give you the insights. But our digital infrastructure tends to be as expansive as our physical infrastructure. And I include our 9-acre greenhouse in that statement when we talk about it. We've basically got a virtual clone of our greenhouse, and we're tracking every single thing as it moves through. And so the data investments we make tend to be about the same amount in terms of dollars as the physical investments that we make as well. And so it's a big part of what we have to do. This probably goes without saying, but good people are the foundation of all of this. Without good people, we may as well not show up to work every day, because everything will be difficult and we won't gain the insights that we need. And so making sure that as a leader, your focus is you're as laser focused on building a good team, creating a good working environment as you are on the technologies that you have is critically important. And then the last one for us that I think, there's different ways we've thought about this over the years, but we're very much in a don't invent what you can't buy world right now. There's a light bulb. I don't need to make a new light bulb. Let me buy that out of a catalog. When you buy things, you get somebody else's quality control for free. You don't have to invent your quality control that goes along with it. You get their tech support, you get their experience. So, there's been times when we've built things that we really shouldn't have, and we spent money hand over fist that would have, in retrospect, been much better invested in supporting business partners, creating better alliances, and working together to solve problems. And so when I think about our greenhouse and the automation that we have inside of it for moving plants around, for moving benches around, 95% of the automation is third-party automation. It's provided by people who've been supplying automation to the horticulture industry for the last 20 years. I don't need to figure out how to move a pot around a greenhouse. That's not a good use of my time and energy, because somebody's on a third generation of that technology already. What nobody's done in a greenhouse so far is grow corn in it, because corn is a field crop. And so where we needed to spend our time and energy was inventing modifications that allowed corn to be really happy inside of the greenhouses. And so from the way we transplant the corn, we had to use slightly, think about transplant differently, to the way we track data. If we're chipping every single seed and we're getting a genetic fingerprint of every single seed, we need to keep track of who's who for its whole life cycle, or we're going to lose all of that investment that we've made. And that's, between 50 and $100 a seed. So it's not insubstantial. Nobody in the horticulture industry cares about that. They tend to think about it, they're doing flats of pansies to sell to Home Depot, or orchids to sell at Trader Joe's. And so they didn't have anything for keeping track of individuals. And then nobody pollinates by and large in the horticulture industry. And so we have to pollinate our corn by hand. And so we had to figure out how to adapt our work environment so that humans could do hand pollination of corn out in our automation area. And so, but we were scrupulously careful to not invent what we couldn't buy. Another good example of this, and you can, I think, Google this and find it on YouTube, but we invented seed chipping technologies, that thing where I talked about we take the chip out of the seed. That's because nobody else, it didn't exist, and nobody could do it. And it's become one of the foundational patents and one of the foundational competitive advantages that Monsanto and now Bayer has enjoyed for the last 20 years when we think about agriculture. And so there is a time and a place for invention, but it's sort of only under duress a lot of the times is when you want to do it. And so these are just a couple of examples. It's a little bit busy, but I wanted to illustrate when I'm talking about scale, what does that look like when we think about genetic testing for us? And so some of these I've been a part of inventing. Some of these were there. I was part of the fourth wave of invention that happened in our genotyping space. So some of this was well before I came along. But what you can see is seed. I talked about seed chipping. Not only do we have seed chipping for corn, but we've got a number of different types of seeds that we built out chipping capability for. This is one person, this is my good friend Mackenzie, running a chipper. that will chip through tens of thousands of samples in a day. When I was a grad student, when I was a postdoc, that was me with a pair of forceps, taking the leaf samples laboriously one by one and putting them in a single test tube. And I was lucky if I could do 200 or 300 a day, partly because of boredom, partly because of the calluses that could only support about 200 or 300 snap tubes a day. This reduces ergonomic strain. This allows us to do more. This is much better at tracking things in a digital way than humans are. So after we've chipped all those seeds, we have to store them. And so this is an automated seed storage. It was once somebody called it Chips Carlton, where we store the seed chips. And so this is where hundreds of thousands of seeds can be stored while we're doing the genetic testing. So after we chip things, we have to keep track of who's who. It's going to take 2 or 3 weeks to get a result back at times. And so we store them until we're ready to offload them and ship them on to our greenhouse. And then lots and lots of automation for DNA extraction and genotyping. So this is where those 4 generations come in: high throughput robotic systems, automated sampling, automated sample tracking, and process monitoring. And so this is a third-party piece of equipment we bought where this piece of tape right here, this little block right here, that holds 364 micro test tubes. And that thing can run through just reams and reams of that tape. It's actually, this is the roll of tape, looks like a film projector in some ways. And that's just the scale at which we're doing the genetic testing as we're running it through a piece of equipment like that. Again, bought off the shelf. We had to custom build this one, so you can see some of the roll frame aluminum here, because nobody knew how to get DNA out of corn in high throughput. And so we had to figure that out ourselves. And we built that in partnership with the same people who built this off-the-shelf piece of equipment. They partnered with us to build this custom piece of equipment for us. And then we had various deals in place to make sure that where we had felt like we had contributed IP, we were able to secure that. All right, so then I think another lesson I've learned over the years is understand what your customer wants. In the case of genetic testing, the things on the left kind of cover what that is. So they could want speed, so a result in 30 minutes instead of 3 weeks. Information density, so instead of looking at 1 spot in a genome, I could look at 3,000 spots in a genome or the whole genome. Low cost, so the more you want to do, the lower you typically want to see that cost. High volume, so millions of samples, hundreds of millions of samples a year. Accuracy, how often is what I'm telling you going to be right? And do you want that distributed so that it's at point of sample, or do you want it centralized like in the last example? And the challenge here, and the thing that I've struggled the most with as a leader in this space, is helping my partners, or we call them partners, I think for a lot of people they're customers, understand what they really want. Particularly because you typically in a new technology can only get 3 of the things in DNA testing on the left-hand side. It's pretty hard to do, to fundamentally change any more than that. And so, but your customer wants all of them. Every single one of these is something they want. And so how do you help them think through what is a fundamental change, what will result in a fundamental shift in the way they approach breeding if we were able to do? And for many, many years, it was high volume, low cost. So it was driving the cost down and the volume up. But then we start to see some shifts in that over the last couple of years, and I can give you some examples of some of that. I think the best one was when speed became an issue. So breeding is driven by generation. So corn takes about 100 days to grow. In the US, we can only grow corn once a year. In other parts of the world, we can grow corn 2 to 3 times a year. But once you're up to 320 to 340 days of just growing time, there's not a lot of testing time baked into that. And our standard was 3 to 4 weeks to get to get turnaround on a genetic test. And so when we did some math, we realized that if we could get 3 generations in instead of 2 generations in a year, that would accelerate our time to market from about 15 years to about 12 years. And so that's huge in breeding. So from 15 to 13, sorry. And so once we understood that was what was going to really change the way we thought about breeding, we focused for a couple of years on how do we make our turnaround time go from 3 weeks to get a result to less than 7 days to get a result. And by getting it down to less than 7 days, we were able to fundamentally change the way we approach breeding. And what was interesting about that is that was a change that was much less about technology, and that was much more about logistics and organization and how we help samples move through a lab. And that's, I think, what we'll pivot to now. So logistics, it's amazing to me how much of science becomes logistics when you're thinking about scale. And so I probably think less about what I learned in college and grad school these days than I do about how do I make sure seed gets to the right place at the right time in the United States. Because if we miss a window, we lose a year. And if we lose a year, then we are a year slower to market, and we lose some, not only some of that market share, but some of the genetic value goes away with that as well. And so that's why a facility like Marana, centralizing this all into one greenhouse, being in protected culture, improves our probability of getting the right amount of seed out at the right time. Again, keeping that acceleration going. The right-hand side, this is a automation line we put in place for handling soy and corn seeds, borrowed predominantly from people who worked in the pill industry. So thinking about counting pills, sorting pills into bottles, et cetera. This is we shifted from where you harvest, you process, and you sort, and you box up, to sending everything we harvest to this one central location. And then they have the leverage, they have the automation and the expertise there to do that efficiently and quickly. They also put those into a format that can be then fed directly into a planter as a cassette, instead of being fed in one by one as envelopes. And so now our planting gets a lot more efficient as well. And so we're able to get our seeds in the ground in days, what would have taken weeks in previous years. And so for us, I think we're at a phase now, and these things come and go in waves, but we're at a phase where logistics is kind of eating everything else for breakfast. We've really got to be really good at logistics. We're starting to hire people with backgrounds in logistics, not backgrounds in breeding or automation even at this point. Another lesson I've learned over the years is it's important to find root causes. My gut instinct is about as good as one might think it is, which is at best 50% accurate. And so using data to make sure that we're understanding what a problem is, not emotion, is incredibly important. I will say what my gut does tell me these days, based on the first line, is maintenance and calibration are usually my biggest problems. So if we're not maintaining our equipment, then we're getting bad answers. And we did actually a really big when we were getting our genotype by sequencing platform up and running. This is moving away from doing some of the simpler approaches to a massively multiplexed DNA sequencing approach, much more sophisticated technology. We had a lot of early success, but we kind of we plateaued out, and we couldn't get any better than about a 5-week turnaround time in that space, and our accuracy was only so-so. And after 3 years of struggling, we were on our second or third generation of technology. We finally commissioned a root cause team to go in and figure out why we were struggling so much. Lots of really smart people working on this, lots of gut instincts, some use of data, but never a comprehensive effort like this. Turned out, and I was leading the maintenance team at the time. It was part of my team, so this is an embarrassing chapter in my life, but it was that I had a maintenance team who liked to fix things, not maintain things. And so we weren't calibrating our instruments, we weren't cleaning them appropriately, and that was leading to most of the problems that we were having. And when you then overlay that on a bunch of other spaces, you can see where that's for us and others I've talked to, one of the biggest problems you're likely to have. However, without data, it's actually pretty hard to know specifically where that problem is. And then I've touched on this, so I won't belabor it, but at scale, particularly when you're trying to get a lot of things done, logistics eats science and technology for breakfast. I don't care how good your idea is. If you're playing the game where you're trying to do lots of things at once, if you don't have logistics, you may as well not show up to work. And it doesn't have to be complicated, sophisticated stuff. It's just making sure you're giving the devil his due, as it will, in that space, and making sure that you're paying attention to those logistics. And one of the interesting ways this showed up was in the way that we thought about pipetting validation. So that's, again, moving those small volumes of liquids. We ended up just adding food coloring to our buffers at a prescribed amount, because cameras can see food coloring, they can't see colorless liquids. And so we had red and we had blue, and then when you mix them together, you get darker red. Turns out a camera can actually see that and can tell us how good we were at pipetting. Now, it's one thing to get a picture, it's another thing to analyze the data, to flag something as being out of whack, and to do all of that. But when you're going fast, when you're going at scale, it's a little bit like a stock market crash that's run wild because of a computer glitch. The problems amplify themselves dramatically, and something that might have taken weeks to solve could lead to tens of millions of samples being incorrect. And so having something flag itself on the first sample, not the last sample, that there's a problem, is incredibly important. And so as we've moved I basically, the other thing I'll tell my teams is I distrust all robots implicitly, not because they are untrustworthy, but because they only do what we tell them to, and we're not very good at thinking that through sometimes. And so I want validation at every step where it makes sense. And that usually needs to be visual validation wherever possible, because things get out of control quickly. I know I want to make sure I leave some time for questions, and this has gone a little longer than I thought. I'll close with some really inspiring advice that the vice president of reading gave me when I started this job. And that was, nothing in your experience has prepared you for this. And I kind of looked at him cross-eyed and questioned why he hired me in the first place, if that's what he thought. But I think this, in this case was referring to the job in Arizona, but this is anything in your life. Wherever you've been doesn't necessarily prepare you for where you're going. And I think that's not a good reason to not do it, and it's not a good reason not to hire someone to do it. But there's a couple of things to really pay attention to, and one is the effective environment. When we move people around, when we put them in new spaces, really talented, clever people express difficulty, or the way that when they're struggling with their job, and the way that having a tough time manifests itself is different by environment, and people perform really differently in different environments. And so understand that sometimes it's not about a person having a hard time, it's about a person in this particular environment struggling. Is there anything we can do about the environment before we start really hammering on the person? Part of that for me is then the second one, which is I believe deeply in experimental leadership. So try something, and if it works, great. This goes back to data, right? So if it works, great, do more of that. If it's not working, let's try something else, and keep iterating until we find something that really works. And then the last one for me, and it's sometimes hard to listen to this advice, but early on, get some good mentors who know a little bit about what you're doing, even if it's a little bit about a part of what you're doing, and get some advice, peer mentors, senior mentors, whoever it might be. However, don't listen to bad advice. Not all advice is really good advice, and so learning to distinguish one from the other is really important. And I think with that, I just will close with saying, for me, it's about the people we work with. And one of the things I love about the project run right now is the vast array of skills and personalities that we've brought to our site in Marana. And it's from all the areas I've shown here, and it's thinking about all the problems I've shown here, but it's the people down below that make this worthwhile, and the problems we're solving at a global scale that get me out of bed every day. So I'll stop there for questions. I always start with what affects a tenfold change in my operation as my base starting point when I'm thinking about scale. So if I want to find 10 times, if I want to be able to do 10 times more, provide 10 times more accuracy, 10 times more something, what's the thing that's keeping me from doing that today? And try to use that as a way to drill really quickly into what is my current bottleneck. And it's sometimes really obvious, and sometimes you don't need to spend a lot of time on it, and sometimes it's a much trickier question.
Thank you.
Thanks, everyone.
All right, bye-bye.